2 of the cutaneous senses, we will continue with the sense of touch. Talk about these two major pathways up the spinal cord for somatosensory information. Both pathways are going to cross over onto the other side at some point on their way to the thalamus. So we have that contralaterality at the level of the thalamus. Most fibers synapse in the ventral posterior nucleus of the thalamus. We also have other thalamic nuclei that receive some information. From the thalamus, we get that information to the primary somatosensory receiving area, or S1, which is in the parietal cortex. And here they usually say we send some of it to the secondary somatosensory cortex, so S2, and that those signals there's communication back and forth between S1 and S2, and then on to those higher level association areas is usually how we talk about anything outside of the primary receiving area. If we look at the mapping or the organization of this structure, the primary somatosensory cortex, we know that we have this kind of body map up in the somatosensory cortex. They call it the homunculus, which means little man. And sometimes you can see some pictures. You can probably just go Google homunculus and find some nice pictures of the little man where they usually put the little man together in a real looking way, as opposed to what we see when we actually look at that body map or that homunculus in the brain, that we kind of have things separated in a funny kind of way where the face is next to the fingers and so forth. But what you'll see in the little man, if you look it up, is the face is bigger and the hands are bigger. We have this cortical magnification, which we'll come back to. If you remember, we saw cortical magnification of the fovea. It meant that there was more representation of the neurons communicating from that region. And so we see a similar kind of thing, and it gives us this acuity, right? Typically, when we are exploring something new and trying to figure out what we're feeling, we do that with our hands as we get older, because that's going to give us that good acute information, or detailed information. Next section is about perceiving details, and so as I promised, we're going to get back to talking about the Merkel receptors, as they're going to give us some of this information about the details of things that we're feeling. And he gives this example of use of Braille. I don't know how many of you have ever tried to read Braille, but it is, for those of us who are sighted and don't need to read Braille, it seems like this impossible task. If you've never done it, you can go to any of the buildings. You can go to, I think, any building in the elevator is going to have Braille. But you can go to any of the buildings at USC and there's Braille outside the stairwells and everywhere. So just kind of feel around and see, is this informative to me? And you can even bring this as a little cheat sheet of what should be raised bumps. But if you think about telling the difference between B and K, it's really just a question of the amount of space, right? And there are others that are similarly, they seem like they would be ambiguous until you get, I don't know, really good at this. People who are blind, we do see, so the visual cortex doesn't just go dark. We don't have these areas of the brain that just end up with no communication to the neurons, in which case the neurons would die. What we see instead is that their visual cortex responds to other sensory modalities, and it appears to be responding when they are reading Braille. So some of that cortical space is devoted to Braille. Several measures for measuring tactile acuity. So one that we can think of is the 2-point threshold, where we're talking about the minimum separation between 2 points on the skin, that if we stimulate those 2 points, we can feel that they are 2 separate points. They don't just feel like 1 point, because if we get them close enough, they might feel like just one large, right? We don't feel that there's a separation in there. And we will see, or you can see, I've done this a few times, if you get 2 toothpicks and you kind of separate them on your finger until you can just feel that there are 2, keep them at that same distance and you move it to your arm and it'll feel like 1. again, and we'll talk about why. Again, we have that acuity, that sensitivity to detail for our hands and especially our fingers. Another measure of tactile acuity is grating acuity. So we looked at these gratings very early on when we talked about vision, and I talked about vertical and horizontal, that we have more neurons and we're more sensitive to vertical and horizontal, that we have this oblique effect. Well, we can take a similar kind of grating and test how narrow or how wide or how big or how much separation there has to be, how much contrast, with just how with the grating of an object that we feel on the skin. Another measure is raised patterns, where oftentimes they'll use letters or something. And I used to, one of those journal articles for the eReserve, I used to have one that was lingual tactile acuity, and they were feeling little letters with their tongues and seeing how small they could get their letters and or what their smallest threshold was, right? And then they compared. They compared older people to younger people and women to men, and just my memory is that we lose some of that acuity as we age, but there wasn't a difference between males and females, but that's filtered through so they can use raised patterns. Receptor mechanisms for perceiving details. It's clearly the firing of the Merkel fibers or the Merkel receptors that are telling us about details. And so here is this bar which has several little things sticking up out of it, and you can see that as we have something that's long, where there's no change, there's no real firing of the Merkel receptors. If there are lots of little ones, there's some firing. We raise the firing. If there's a little one and it's really separated so that we can tell, right, that there's one thing sticking up, then we'll have the most. The Merkel receptors will fire the most. If we then look over at what's happening with the Pacinian corpuscles, first of all, you'll notice that for the Merkel receptors, we're going from 0 impulses per second to 100 impulses per second. So those ones that are those large changes, those neurons are firing at like 100 impulses per second to say, hey, I feel something here. Whereas if we look over at the Pacinian corpuscles, we're going from about 5 impulses per second to about 20, maybe a little bit over 20. So they're not firing very much. And it is obvious that it doesn't really matter, right? Whether this is a longer piece of bar, whether there's something narrow that's sticking up, whether there's something narrow sticking up and it's by itself and not with a few others. The Pacinian corpuscles, they aren't, the firing rate isn't changing that much, so it isn't telling us about that detail. Receptor mechanisms, or the mechanisms of what gives us better acuity. Better acuity is associated with, first of all, well, a number of things. So first of all, less spacing between the Merkel receptors. So you can see in the figure, as I'm getting more space between the Merkel receptors on the x-axis, I also need those grating acuities to be bigger for me to be able to feel the direction of the grating. And this makes sense if we think about also how the fovea was tightly packed with cones, so there are more of them in there, and we can think of that as less space because the retina is a spatiotopic kind of map. But same with our fingers. So as we get those Merkel receptors closer to each other, and we might have one firing and then the ones between them not firing, and then another one firing, we can tell, right, that there's that space between them. This matches up nicely with our 2-point thresholds. So the lower the threshold, the more sensitivity, right? And you can see there's more sensitivity on the face and there's more sensitivity on the hands. So those low bars. Whereas when we get to the arms or the legs, there's we have worse acuity. We have higher thresholds. Second mechanism that is associated with better acuity is a neural kind of mechanism, which is the receptive fields. So these areas on the skin, when stimulated, that then influence the firing rate of a particular neuron. So it's always what are the receptors that are affecting the firing rate of a particular neuron, my receptive fields, but in this case, so it's the area on the skin. And if we look at these receptive fields, we can see that there are small receptive fields on the fingers, and they are less overlapping. As we get up to the palm, and especially if we compare that to the arm, those receptive fields are larger and they're overlapping with each other. So if we think of this as kind of the rods and the cones are in our fingers and the rods are in our arms, if we make that comparison, we can again see, so if I separate out those points in A on the fingers, I have a neuron firing. I have two neurons in between that are not firing, and then I have another neuron firing, and so I can tell, hey, these are two separate points. But I pull that same distance up to the arm, and what's happening is there are neurons firing and there's overlap, right? Some of the same neurons are firing, and so it's not as clear. I need to pull those two points even farther apart. I'm going to raise my threshold. The third mechanism that gives us this better acuity or more sensitivity to detail, and again, this all should feel like, hey, this sounds like what we were talking about with vision, is the cortical magnification. So the fact that we have that cortical magnification of the face and of the hand, that is where we really have our best acuity with the tongue, aspects of the face. And I often do this, so being able to feel our faces and move our faces and our tongues and our articulators very seamlessly is what helps. But language is complicated. We're doing a lot of movement and feeling as we are creating these words. If we want to think about perceiving vibrations, which we're actually going to talk about this as helping us to potentially perceive details when we're exploring something with our hands or however we're exploring things, the Pacinian corpuscles help us to perceive vibrations. And in what is now a really classic study by Werner Lowenstein in 1960, so I often compare this to a waterbed, where if I get on, you feel it happening, and if I get back off the waterbed, you feel it happening. And so if I have a kind of vibration, it's going to cause this movement, right? And that's what we see with the Pacinian corpuscles. And so one of the things that Lowenstein did was he did a continuous pressure out at the outer edge of that corpuscle, where the corpuscle piece you can see kind of looks like an onion. So we have these layers with liquid in between them, and in the very center is a bare nerve ending, which bare nerve endings are what we're going to end up seeing when we talk about our nociceptors and our pain signals. But so, this is very protected, right, by this oniony layered thing. So if I have continuous movement or continuous pressure out there, it feels it when it starts, and it feels it when it stops. And he shows in section C, if I push at A up there, again, I'm going to feel it when it starts, I'm going to feel it when it stops. But then Lowenstein went and he peeled away the onion and he pushed at B, right, or stimulated at B. And now there is a continuous signal. So it is that oniony structure and the fact that that liquid is moving and then stimulating that bare nerve ending. That's what gives us this perception of vibration. If we look at the history of this field of perceiving by touch and perceiving texture, most of the research has been done on spatial cues. If you can see that in 1925, David Katz distinguished between spatial cues and temporal cues, where temporal is, again, through time. Those spatial cues, when I can feel the relatively large bumps and grooves on some kind of surface. And I usually, in front of the class, take my wire ring notebook and push it up against my hand, right? So some of those, and you can do this, put a wire ring notebook up against your hand. Some of those Merkel receptors are going to get that sense of pressure and they're going to fire. There are going to be ones in between that are not feeling pressure. And so this is a spatial kind of cue. It is happening through space. And so when we are pushing elements onto our hands or pushing our hands against certain elements or certain things we want to recognize and feel. Temporal cues are happening over time, and they happen because of the vibration that can occur when we are moving over the surface of something that's coarse, like sandpaper, or we'll look at some different sizes of grains of sand, basically. So it's going to tell us about texture as well. And so Mark Hollins ended up calling this the duplex theory of texture perception, and Hollins and his colleagues have done a lot more research recently on those temporal cues to tell us about how we're receiving those temporal cues. I'm just going to go through one of these examples of the research from Hollins's lab. So Hollins and Reisner in 2000 did this can you feel the difference, or what is the difference that you feel if you're making this kind of magnitude estimation? How different are they between 10 micrometers and 100 micrometers? So sand that is 10 micrometers large or 100 micrometers large. And you can see that the sand that is 100 micrometers is going to be 10 times as large, right? So, but if what they did was this static, so all I'm doing is just pushing my hand in and I'm just getting those spatial cues. If that's all I'm allowed to do, that you can see that people were rating the 10 micrometers, the very fine, and the 100 micrometers, the fine sand, as being, the 100 felt larger, but not, they're kind of felt similar, according to this figure. If they were allowed to move, so to use those Pacinian corpuscles, then we can see that gets distinguished to be almost 10 times as large now. I can tell that this one is fine, and the 10 micrometers in comparison is very fine. Another way we can perceive texture is through vibrations using a tool. And so if you want to do the same kind of thing with your wire-edged notebook, so if you press it up against your hand, you're getting spatial cues. If you're moving your hand back and forth, you get some temporal cues, which they say is still Merkel receptors because it's not fast enough, but I'm curious, it's still a temporal cue and cerebellum's still involved, so I don't really know how much I don't know how to, I feel like those Pacinian corpuscles are still must be involved, but that's not what the research says. And then if you now take that wiring notebook and you take a pen or a pencil and you feel, you'll notice that what's interesting now is what you're sensing is this vibration of the tool itself, of the pen or pencil, that you're interpreting that as the texture of the wire-ringed notebook. I do this distinction between active touch and passive touch, where active touch is where we are actively exploring an object, where we usually are using our fingers and our hands and we're moving them around in this kind of haptic exploration of these 3D objects. And we'll look at some exploratory procedures that they've discovered. And I will give this example of active touch from the textbook that I like a lot. I'm going to say the words, the name's wrong. So Girat Vermeij, I feel like I'm making that up. But blind since the age of 4, was being interviewed for getting into Yale's biology department, so a very prestigious department of biology. And of course, they're giving him this little test. He's blind. They're figuring, hey, he's going to fail this test. And so let's just do this. And so here's the description on page 368. Here's something. Do you know what it is? Bowl asked as he handed me a specimen. My fingers and mine raced. Widely separated ribs, parallel to outer lip, large aperture, low spire, glossy, ribs reflected backwards. It's a harpa, I replied tentatively. It must be a harpa major. Right so far. How about this one? Inquired Bowl, as another fine shell changed hands. Smooth, sleek, channeled suture, narrow opening, could be any olive. It's an olive. I'm pretty sure it's Oliva cyana, the common one from Florida, but they all look alike. Both men were momentarily speechless. They had planned this little exercise all along to call my bluff. Now that I had passed, Bull had undergone an instant metamorphosis. Beaming with enthusiasm and warmth, he promised me his full support. And of course now, Girat Girat Vermeij is at the University of California, Davis, Distinguished Professor of Marine Ecology and Paleoecology. So really great example of active exploration, and of course most of us would not do that well with shells, but he obviously, especially just through feel and without our sense of sight, but he obviously was a professional. He had worked with these shells for a long time to learn this, and he is blind, so this active sense of touch is very informative. Again, we often distinguish this from passive touch, where someone's maybe putting something on the skin, and even the way we talk about whether something feels like it is sharp or it has ridges, which is more active, versus passive touch, where it's sharp and it's sticking me. We talk about it as more of something that's happening to my skin as opposed to describing the object when we're having that kind of active touch. About what's happening with this haptic exploration, we are using these various systems together. The sensory system, where we're feeling what we're touching, whether that is there's ridges, the texture, temperature, that information. We're using the motor system because we're moving our hands, fingers around something, and we're using our cognitive system, where it is receiving information from these systems and sending information right to the motor system based on both where I moved my hands last, but also what I'm feeling. And so this is a really quite complicated use of and integration of these various systems. When we get to that thalamic nucleus, the ventral posterior nucleus of the thalamus, we have center surround receptive fields. This should sound familiar, right? And I may have shown this picture when we talked about center surround receptive fields for vision, because for vision, it seems really abstract, and for touch, it makes some amount of sense. And so I did this thing, or I have been doing this thing, so I hope I did it in this class, of really touching yourself with your finger, where it's a little point. And so we're going to be hitting right in that in a center, right? And as you get larger and you start to hit some of that surround, and you have those overlapping center surround receptive fields, what you really notice so if you put your whole hand on your arm, what you really notice is where that touch begins and ends, but that particular center where you might have just been touching with your finger, it isn't as noticeable now. So this is the point of lateral inhibition and these center surround kind of receptive fields. It is to distinguish where things change. And a good way to visualize this, I think, is this picture that I'm not sure is in the book anymore, but so if I'm touching a small ball with my finger, because I'm hitting those center surround receptive fields and I'm hitting just the ones kind of in the center, I'm getting more center. We're going to see this sharp incline in the amount of neural firing, and then this sharp decline, right, where the neurons around that aren't firing so much. If I'm touching my finger to a larger ball, so that really it's probably much more of my finger, maybe my whole finger is touching that ball, what we see instead is there isn't that sharp incline or sharp decline, because there's more neurons firing, but the pattern of firing is telling me that this ball is more shallowly sloped. Add just a tiny bit of touch to the pain chapters, because this is getting a little long, but I wanted to do one final concept here, because this really feels like it belongs all together. As we're perceiving objects, we get that information past the thalamus, which we just talked about having center surround receptive fields, up to the cortex to S1, our primary receiving area for touch. We're still going to see some neurons have center surround receptive fields, but other neurons are going to respond to more specialized stimulation. And so what this picture is showing, so if we go into that homunculus, and so that's my surface structure, right, and I go in a certain area, and I go in perpendicular and start measuring, we're going to see neurons at a certain point that are responding to that area of the hand that are also responding best to horizontal bars hitting that area of the hand. not so much response to oblique, really no response to vertical, because I'm hitting some of that. I'm hitting more of the inhibitory surround than I am the center. And so we see these, I'm going to end up telling you what this reminds you of. We see these things like simple cells, right? They sound like simple cells in the visual cortex. And then neuron 2, or the second example that he gives here, it's a neuron that is responding best, not just to being stimulated on the finger in some way, but being stimulated by an oblique bar that's moving up and to the left. It doesn't respond if something's moving down and to the right. It's only when it's moving up and to the left. So this starts to sound like those complex cells. So somatosensory cortex has similar kind of organization because it's spatiotopic with some cortical magnification of where I want my better acuity or better sensitivity to detail. And with the receptive fields, we're responding to features that are similar kind of to our retinal features because, again, we're maintaining information about space. So it's going to be lines of a particular orientation or moving lines, lines moving in a certain direction, like that. Also in primary somatosensory cortex, we can see that neurons respond best to specific objects, but you can kind of think of that ruler as here's a sharp line, right? So it's still sort of feature-like, but it's also has to do with that object because if we have that shallower slope in this kind of cylinder, that specific neuron that responds to the ruler or the sharp line is not going to respond. So they can tell us about objects at this level.